I'll move on to talk about some of the combination trials that are ongoing now. This is the most, I don't know if straightforward is quite the right word, but it's the most obvious extrapolation from some of the remarkably promising results that we saw in melanoma. When patients were treated with both the CTLA-4 agent ipilimumab with nivolumab, and we saw some of those curves before where these patients have responded remarkably well. And as Dr. Weiss showed, that some of the results initially in lung cancer have not been quite as promising. I don't think that's a dead end, but I think understanding how best to use these therapies together in safe ways with right doses in the right patients is a critical challenge. And I think the opportunity to use two drugs together that have different mechanisms of action, that have the opportunity to work synergistically together is important. And so there's at least two trials, one that's sponsored by BMS and one that's sponsored by MedImmune, and there's another one coming with Genentech's therapy that are asking this question of how best do we combine these two therapies together, knowing that in melanoma that was a game changer. Another sort of combination are not two immunotherapies together, but standard therapies plus immunotherapies. I think this is a hugely important open question of can the treatments that we use now prime the immune system to work better? And that includes changes to the tumor as well as the environment that surrounds the tumor. We know that the tumor wants to invade an immune response, and it makes things that prohibit the immune system from responding well. Chemotherapy may also change that microenvironment to help immunotherapies work better. Thus far, as you saw, that the results weren't astounding from what's been described thus far. But I think that the opportunity to use standard medicines together with immunotherapies is going to be incredibly important. And I think that this can work, but understanding in whom it's best to work and what medicines may work best together, it will be an important part of the future. This is a small study, but is an example of how I think rationally we can start to think about what makes immunotherapies work. We've talked a little bit about this morning that what makes a cancer recognizable to the immune system are those antigens on the surface of a cancer cell that makes it look different, that makes it look foreign, that can make it look like a bacteria or a virus. And there may be multiple ways in which we think about making that cancer more obvious to the immune system. Vaccines are one of those things to show the immune system what that cancer might look like. But this is another example in which we focus on something called epigenetics, which is where the cancer may actually suppress the expression of a specific antigen. And that by giving this epigenetic therapy, you force the cancer to re-express that antigen. So something that's hidden is now suddenly shown. And that the combination of the epigenetic therapy plus the immunotherapy may work well. And the group at Johns Hopkins had shown that, incidentally, though they weren't looking for it initially, that patients who got this epigenetic therapy and happened to go on to an immunotherapy trial later, some of those patients did really well. So 3 of the 5 patients had a tremendous response. And so the question is, can this epigenetic therapy actually change the way that the tumor looks to the immune system? And so that's the point of this study here. This is a sort of a different type of combination therapy. And we know that lung cancers, we've probably seen those pie charts where lung cancers are broken up into multiple different pies, with each of the slice of the pie being a specific mutation within those cancers. That's EGFR mutations, that's KRAS mutations, et cetera. And we've historically, or at least in the last decade or so, thought of those cancers as being distinctly different, and they probably are. But they also have targeted therapies that can potentially work in some of them, EGFR-directed therapies, ALK-directed therapies, et cetera. And that was the huge benefit of the mid-2000s, of finding molecularly targeted therapies. I think today we're seeing the new wave of types of treatments that build upon the success of molecularly targeted therapies. But we should try to figure out how to put those together, molecularly targeted therapies with immunotherapies, rather than ignore the successes of the previous decade. And this is one of several in which patients with metastatic EGFR mutant lung cancers are treated with either erlotinib or Tarceva in addition to anti-PD-L1 therapy. Let's see if this is the next slide. Yeah. So the difference between these two This is for patients that have just been diagnosed with EGFR mutant lung cancer. They haven't had any therapy yet. And by giving 2 drugs together, up front, the idea is, can we circumvent resistance or can we delay resistance, which otherwise is inevitable in EGFR mutant patients? The second study is looking at the time in which they have already developed acquired resistance and can either PD-L1 drug on its own or in combination with these 3rd-generation EGFR inhibitors actually improve the tumor at the time of acquired resistance. I think both approaches are important, and I don't know what's best, whether it's best to give everything you've got up front or to be able to do things in sequence. And both of these studies, I think, will help us ultimately answer that question. KRAS is the most common type of mutation that's found in lung cancers, but it's one that's been incredibly difficult to try and target. It was, I think, identified in the 70s and 80s, and it's been several decades now without an active therapy. I think this is one study that's trying to do better. We know that KRAS and other specific mutations signal down a pathway that teaches a cancer to grow and divide. And one of the specific molecules along that pathway is something called MEK. And so this trial, particularly for patients with KRAS mutant lung cancers, is combining a MEK inhibitor with a PD-L1 agent. And so this is, again, the idea of how do we shut down the growth of the cancer while also amplifying the immune response to that cancer. And touch briefly a little bit on some of the things that are beyond PD-1 and PD-L1, though many of them are sort of building upon that platform, and that makes sense. We have, I think, surprising but remarkable efficacy with the PD-1, PD-L1 drugs, and the idea is how do we do better? And rather than sort of reinvent the wheel, you can understand why people would start to build upon it rather than start from scratch. I have a list at the end of some of the other targets beyond PD-1 and PD-L1, but this is one of them that's in clinical trials now of a drug that's targeting a CD137. We've talked about the idea of accelerators and brakes in the immune system, and that PD-1, CTLA-4 target the brakes. By taking it away, hopefully the immune response to cancer can amplify. But there are also ways of pressing on the accelerator, and CD137 is one of them, though not the only one, in which you can play on both sides of the balance. And hopefully by amplifying the immune response, it can target cancer. This is another idea where in the immune system, there's multiple different types of cells. We've talked about T cells primarily today, and that makes sense. But there are other cells in the immune system that can respond to cancer. And one of them is NK cells. These are parts of your innate immune system that Dr. Pardell talked about this morning, and that there are ways in which you can amplify an NK cell response, hopefully to cancers as well. And so this is one of those examples, though it certainly is only the beginning of trying to combine the adaptive immune response with the innate immune response to cancer. LAG3 and TIM3, I suspect, are other molecules that you'll hear a lot about in the next coming years. And these are molecules that are very similar to PD-1. They upregulate in the context of activation of T cells, and they're part of what makes a T cell not respond as well to a cancer, that causes a T cell response to become exhausted. And just like with PD-1, by targeting LAG3 or TIM3, we may be able to reestablish and reinvigorate that immune response to cancer. And so this trial now, which is a phase 1 study, it's developed by BMS, is a LAG3 antibody with another arm that's combining the LAG3 with their PD-1 drug, nivolumab. We should say that the majority of our discussions thus far have been around non-small cell lung cancer. But there's no reason it has to be. We've seen how immunotherapies have transcended the idea of cancer subtypes. That the idea that the immune system matters in cancer is an idea that is relevant across multiple cancers, and that includes small cell lung cancer. It includes potentially any type of cancer. But it's only beginning to be explored in small cell lung cancer. I have no reason to think that it won't work. And this is one example of a study that's being sponsored by BMS in which the CTLA-4 and PD-1 drugs are given together for patients with small cell lung cancer. Beyond PD-1 and PD-L1 is a tremendous array, and I think will be a hugely important part of our future. The fact that the drugs that we've started to look in so far work is remarkable, almost surprising. Actually, it is surprising. But they do work, and it's an important proof of principle that this is a system that can be harnessed to treat cancers. But it's only one way. It's only one way that a cancer evades an immune response, and it's only one way of medicines that are able to reestablish that immune response. And there's a diverse array of medicines, of molecules, of therapies and strategies and approaches that I think can be effective in treatment of not just lung cancer, but any cancers. So these are sort of concepts, drugs, and ideas to keep in mind as we move forward from here. I suspect that all of these will be important parts of our future in treating patients with lung cancer. So to summarize, there's a lot. And there should be. It's very, very exciting. But there will be more as well. And this is really just the beginning. What all of this has shown is that immunotherapies can work for patients with lung cancer. And it's critical that as patients and providers that we work together to make sure that whether it's a clinical trial or standard therapy, or as these immunotherapies become standard, that we identify what are the best options for patients. And that includes trying to find prediction tools for both safety and efficacy, as well as understand the needs and desires of individual patients in order to help them accomplish what it is that they want. And that's it. So thank you, guys.